Hello everyone and welcome to our summary on microscopes. What we're going to be doing in this video then is having a look at a couple of the statements from your specification. 2.1.1a, which is having a look at how we use microscopes in order to observe and investigate different types of cell and their structures. And then we're also going to look at 2.1.1f, which is the difference between two key terms, magnification and resolution. First thing then is we're going to jump straight into those two key terms. In theory, these are phrases that you should know from GCSE. We have magnification and we have resolution. When we talk about magnification, this is just the number of times larger that the image appears compared to the actual size of the object. So we generally refer to these as a times something. So you might see times 100, for example, which just tells us that when we're looking at the image, that's 100 times larger than the actual object we're looking at. The second term we need to know is resolution, and this is your ability to see more detail in an image. The one that you probably learnt at GCSE was something along the lines of the smallest distance between 2 points, where we can still see them as separate points. And that's still true when we get to A level, it doesn't change. We are looking at obviously more detail in an image, and the reason that we can see more detail in the image is because we can actually see objects that are close together as separate objects. So the smallest distance between 2 points where they can still be seen as separate objects, that is the resolution. And different microscopes have different resolutions. What we're going to do then is have a look at 4 different types of microscope that you need to be familiar with. You get a range of questions on the exam papers about these. Multiple choice comes up quite frequently, asking you to identify the correct row from the type of microscope using resolution and magnification facts. You also get some of the short answer questions regarding key things about said microscopes. So what we're going to make sure we can do is obviously recall these key facts so that we can answer all those different variations of question. The first microscope then are light microscopes. These are the ones that should be very familiar to you. They're the ones that you've been using since year 7. In terms of their magnification, these are the lowest magnification. They're just times 1500 as a maximum. Resolution, 200 nanometers. Now, what we find is that the resolution of our light microscope is actually quite limited for the simple fact that we're using visible light in order to actually obtain this image. Now, visible light itself has wavelengths between 400 and 700 nanometers. And what we say to work out your resolution is from the shortest wavelength, which will be 400, basically you won't be able to see something half that. Okay, so hence 200 nanometers, because if our smallest wavelength is 400 nanometers, divide that by 2 and then we get our 200 nanometers. So that's why the resolution is 200 nanometers, because it's half of the smallest wavelength of our visible light that we use. What we've got here then is just that hopefully very familiar diagram of a light microscope. Now, I'm hoping we all recognise all the key parts on here, and we should know how to use these, because that is of course one of your assessed practicals that you should have done as part of your practical component. Now, what we do have on our light microscope are 2 different types of lens. We have the eyepiece lens, which is up the top here, and on this one we've got 2. The ones you tend to have in school tend to only have a single eyepiece lens, but it's still an eyepiece lens. We then have our little nose piece, which has our array of objective lenses. Now, the objective lens is the one that you can twist round to change to different magnifications, basically. So we'll have an eyepiece, which is usually times 10, and then our objective lenses, the ones you've probably got on your school microscopes, you've probably got a times 4, a times 10, and a times 40, usually. They can have different ones, and particularly if you are doing things that require you to see a greater magnification, then we can obviously have more powerful objective lenses on there. The little bit that we put our microscope slide on is the stage, and hopefully you've got some decent A-level microscopes which allow you to have some little controls to move that stage around. If you've still got some of the more basic models, let's say, then you've got no controls for the stage. You can literally just obviously move it up and down. You can't move it side to side and up and then forward and back. The A-level microscopes, which hopefully you've had, allow you to move the stage around, because that means that you can then get your image where you want it to be without trying to maneuver a slide, which always ends badly. We then have this little thing here called the diaphragm, and the diaphragm is all to do with changing the amount of light that's passing through our specimen. We've got our light source underneath, and again, if you've got more modern microscopes, that will be an actual light fitting at the bottom. If you've got some very archaic design things like what I had when I was at school, it's a dodgy mirror, probably held on by a bit of blue tack by this point. Obviously, the fixed light source, way better. We then have our two different focusing dials on the side here, the fine focus for that fine tuning, coarse focus for much greater movement. The first calculation that we should just remind ourselves of is to work out our total magnification. Now this is by far the simplest of all calculations, because all we need to do, whatever the eyepiece lens is, times the objective lens that we're using, gives us our total magnification. So if we had an eyepiece which was times 10, our objective lens was a times 4, then 10 times 4 gives us a total magnification of times 40. It's that straightforward to do. If they gave you a total magnification and said that it was times 100 as the total and the eyepiece was times 10, then all we would do is 100 divided by the 10, which gives you times 10 for your objective. So really straightforward to use that calculation just to get the total magnification from those two types of lens we use. What are some advantages of light microscopes then? Why do we still use these things if they've got a relatively limited magnification and resolution? Well, first of all, they're cheap. Okay, you can pick up a light microscope for under £100. Now, that obviously means they're accessible to a large number of people. They're easy to use. You can prepare the slides yourself. You did that in year 7 even. And they're portable. So we're not destined to do all of our experiments in a lab setting. You can literally take that light microscope out into the field. And if you have the opportunity to do some of this biology field work, then particularly if you get involved in any of these expeditions that go on overseas that you can do, you will be using a light microscope in the depths of a cloud forest somewhere in Honduras, for example. We can also study whole living specimens. Things don't have to be dead when we're using a light microscope, so that's really beneficial to us as well. The second type of microscope is the laser scanning confocal microscope, the delightful mouthful. Now, in terms of our key facts on this one, in order to use our laser scanning confocal microscope, then we need to stain our cells with these fluorescent dyes first of all. Now, once we've stained them, what we're going to do is use a laser beam, hence the laser scanning, and we literally scan the surface of our specimen. Where the laser hits some of these fluorescent dyes, it reflects. Now, that allows us to generate this colour image. What are some of the other key facts about this? Well, we can observe whole living specimens and we can observe individual cells in this way. So it's quite flexible in that regard. It also has a high resolution and a high contrast. So these are a couple of key facts that make our laser scanning confocal microscope potentially a better fit for some of our work than the light microscope. In terms of the advantages of our laser scanning, then we can use it on thick specimens, so we don't have to have thin slices, and we can use it on three-dimensional specimens. We also can produce these high-resolution three-dimensional images. Remember what we said about resolution? That's all to do with giving us this real clear image so that we can see a lot more detail. So because we've got high resolution, that means we can see a lot of detail in 3 dimensions of what we were looking at. There are, of course, a couple of disadvantages to be aware of. Firstly, this whole thing is a slow process. So in order to get that image, it's going to take quite some time. And in the process of doing so, we can damage the cells themselves. Third type of microscope is the transmission electron microscope, or TEM for short. Now, in terms of the transmission electron microscope, rather than using light of any form, as the name of this one suggests, we are using a beam of electrons. Now, before we can shoot electrons in a beam at our specimen, we need some very important conditions. We need to make sure that our specimen, so whatever we're looking at, has been chemically fixed. And by that, what we mean is that we've dehydrated it, so we've removed any water from the substance, and we've stained it. Now, obviously, in the process of dehydration, we've got to kill the sample, and it's only because if we weren't to dehydrate this, when we actually put it into our electron microscope, these electrons, they're very tiny. If there was a water molecule present, it would cause deflection of it, and therefore we get a blurred image. So that's why we need to dehydrate. What do we get at the end? We get a two-dimensional black and white image. But here's the really good thing. This is a magnification of up to 500,000 times. That's a lot. So this means that we can see really tiny structures within the cell. And it's got a resolution, you can see there, of 0.05 to 2 nanometers. So again, it's not just that we can see things large, but we can also see things incredibly clearly. Those bits that are very close together, we can still see as those separate objects. So we've got this very high magnification and resolution here, which makes our transmission electron microscope something that has revealed a lot about subcellular structures. Advantages then, we've kind of touched on this. They've got high resolution images first of all, and therefore we can see these internal structures. Things like ribosomes that we can't see with a light microscope because they're too small. We can see them with the transmission electron microscope. Disadvantages, specimens have got to be dead. Obviously if we dehydrate something, it is not living anymore. So the specimens must be dead, and when we're actually preparing these specimens, we can generate something called an artifact. Now, artifacts quite simply are structures that we form as a result of this preservation and staining process. So they're not actually bits of the specimen we're looking at, but when you then look at the image, you might think, oh, that's something in there. It's not, it's just something we've created due to the process we've put that specimen through. And we don't have a colour image here, it's only black and white. Our final type of microscope, the scanning electron microscope then. First key fact, this is the one that creates three-dimensional images of the surface. So we're still using electrons, hence the name electron microscope there, but what it does is it actually scans them across the surface of whatever we're looking at, and from that we then build up this three-dimensional picture of the surface of that object. Resolution, not quite as good as our transmission electron microscope. You can see 5 to 50 nanometers this time. Magnification, again, a bit less than the transmission electron microscope, times 200,000 here, but the images are still in black and white. So we do have some key differences between our scanning and transmission electron microscopes. Your transmission electron microscope is the one that has the greatest magnification and resolution there, whereas your scanning electron microscope gives you a three-dimensional image of a surface. So two different uses for the different types of electron microscope there. In terms of the advantages of our scanning electron, we get three-dimensional images formed from a three-dimensional specimen. We can see what the surface of a ciliated epithelium looks like with this. Disadvantages, has a lower resolution than the transmission. We can't observe live specimens still. And as with the transmission electron microscope, it's expensive. We're talking about something that's going to be firing beams of electrons again. This is not cheap. Also, neither of these are portable, folks. You're not going to be wheeling out a transmission electron microscope or a scanning electron microscope into the depths of a forest. So these have to be carried out on site in a lab. As always, don't forget to subscribe to the channel so you can see when another video is uploaded, and head on over to the website for A-Level Biology, where you can find a whole array of different resources to hopefully help you in your A-Level studies.